Hey guys, welcome back for translation. There's one thing I forgot to add on this. I forgot to call this mature RNA. So this is now our mature mRNA. It can now exit the nucleus through a pore. Okay, so our messenger RNA is created, it is mature, and now it will travel out of the nucleus either to the ER or to a free ribosome. What we're gonna draw for translation is we're gonna draw it at a free ribosome just because it's a little easy. So flip your pagina. Let's title this buddy Translation. Okay. So before, how we kind of like rewrote our DNA as an RNA message, now we're going to take that RNA and we're just going to, instead of just like rewriting it, we're going to completely change its message and we're going to turn it into the language of a protein. So kind of think about here in transcription, we stayed in the same like language, quote unquote, the language of nucleic acid. Okay, we just rewrote it. Over here, we're going to completely change the language. So we're going to take mRNA and we're going to create, let's say use mRNA. Sorry, my sentence, terrible. Use mRNA to create a protein. So we're going to create this whole new molecule just using the instructions from mRNA. So think about this like if I was trying to teach you, my cat just sneezed. If I was trying to teach you how to make a chocolate chip cookie, I would tell you how to make the cookie. So that's the mRNA, the message, and you would create the cookie. The cookie is the protein. So the product of these instructions is our protein here. Cool. I am telling you right now, this is my least favorite diagram of the whole year, only because I haven't really found a way to make it organized yet. It's still just kind of like a hot mess diagram, but we're gonna be okay with that today. Okay, let's start with our hot mess. So this is all cytoplasm, so let's kind of maybe add some directional terms. This is cytoplasm. I'm not going to draw a nucleus, so we're kind of just picking up right where we left off over on this last page. We're going to pick off down here where our mRNA has left the nucleus. Okay? So, you're going to draw your mRNA strand. Let's just draw it a little longer than on our last diagrams, just so we have space to add everything. We're going to have our bases. Let's see. I'm going to draw these a little closer together. You want a multiple of 3, if you didn't notice I was doing that. On our mature mRNA, we have our 5' cap still. This is my 5' end. My 3' end still has that poly A tail. Right? But there's a ribonuclease down here and it's just chopping those off one at a time. So hopefully we can make our proteins before that ribonuclease gets to our actual coding sequence. Cool. So on mRNA, when I'm talking about this code, these instructions to make the protein, if I were to teach you how to make a chocolate chip cookie, I would put those instructions in the form of words. So I would group my letters into like a really specific order, and each grouping of letters has a meaning. mRNA does the same thing. It's going to group its letters in what we call triplets. So every 3 are going to code for a different amino acid. Okay, this is called a codon. So a codon is an mRNA triplet. Wonderful. At the beginning of every single mRNA strand, they start with a really specific codon. It's called the start codon, and it signals the ribosome that this is the beginning, along with the 5' cap. This is the beginning of the strand instead of the ribosome like chomping on down here. So the start codon for every single protein is A, adenine, uracil, guanine. So that's found at the beginning of every single RNA, which means that the complementary DNA, TAC, is found at the beginning of every single gene, right? So that kind of helps the polymerase determine where the start of a gene is. At the end of every mRNA, this last codon, we call the stop codon. And there's 3 different ones that could code for it. It's UAA, UGA, or UAG. And I just remember it because I go ooh-ah-ah, ooga, ooh-ag, because I'm strange like that. You don't need to memorize any of these sequences. It's just kind of nice to know. So let's just pick my favorite, ooh-ah-ah. So uracil, adenine, adenine, that just codes and tells the ribosome, this is the end of the gene, you stop. Let's say that ribonuclease is just chomping, 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 and then it chomps into our stop codon. If the ribosome never detects a UAA, then the ribosome wouldn't ever stop. It would have to stop eventually, obviously, because it's going to reach the end, but you would never have that final amino acid, that stop codon, on your protein. Wonderful. Okay. So that's kind of just going over language stuff. Next, in the cytoplasm, free floating, you have a new type of RNA, and they're called tRNAs. So I'm just going to draw a bunch of tRNAs out here in the cytoplasm. tRNAs are shaped like capital T's. So I usually just draw them like this. On one side of the tRNA, they're going to have a sequence of 3 RNAs, and on the other side, they're going to be carrying an amino acid. It looks like a really weird, sad person. I know. Okay. Sorry, let me search tRNA for you really quickly so I can show it to you. Didn't have it ready, I'm sorry. That's not a good one. That one's crazy. I want it bigger. Bigger. This is fun. Bigger. Bigger. Okay. Oh, you're about to go on an adventure, and I hope I don't drop you like I did that one time. Oh, I did it. Okay. So this is what tRNA looks like. So you can see here, it is shaped like a little T. It's just a strand of RNA. So we just have the A's, U's, C's, and G's. But remember from when we talked about the history of life, how RNA can fold and bond with itself. So this is a strand of RNA that's folded and it's bonded up on itself. Down here are those 3, what did I call them? Just like RNA nucleotides that are found at the bottom of our tRNA, and it's what we call the anticodon. And then we'll have up here, this is the amino acid, or like the little head of our little creepy stick figure we drew. Cute. So that's essentially what we have here. So down here, this is like the bottom of that T loop where we have these 3 nucleotides, and then up here will be its complementary protein. So let's just draw the one that complements AUG, right? So A pairs with U pairs with A, G pairs with C. And the start codon for every single protein is methionine, or we designate that MET, MET. That's hard to read. Let me go over that with pen. Okay, so like this tRNA will have a slightly different sequence from another tRNA, and then it allows it to carry methionine. Let's say there's another tRNA over here. One side will carry its amino acid, one side will have its triplet. Sorry, this triplet is called the anticodon, now you know. It's called the anticodon because it's opposite the codon. So this is a tRNA triplet. Okay. So let's do this. Let's fill out a sequence here, and then we can make our tRNAs that complement. So let's do AUG, and then let's just do U, and then ACG, CUG. Let's just do that many, but understand there will be more. Do you just want to do it? Let's just do it. Ooh, do not do ooh, ah, ooh, ga, or ooh, ag. Yak. Ag. Cute. Okay, so essentially, these are going to get read in thirds, so triplets, like so. So we just break up our codons. For every codon, there will be a tRNA that complements it. So every codon represents an amino acid. Let me zoom out a little. So if I have 1, 2, 3, 4, 5, 6, 7 codons, the stop one doesn't count, it doesn't add an amino acid. If I have 7 codons, that means this protein is going to be 7 amino acids long. That means I need 7 tRNAs. Okay, so let's just draw a few. I don't want to draw all 7. It's going to get insane because this might be the diagram that gets me organized for this lesson. So let's draw 4. Oh, this one really looks like a person. Cute. A pitchfork person. Okay, so let's just kind of draw them in order. So U would pair with AAA. ACG would pair with UGC. CUG would be GAC. Cute. Now you might be asking yourself, how do we know the amino acids? This is when you have to use what's called the codon table. Wow, jiggle jiggle. Or the codon wheel. Here we go, another adventure. Don't drop the phone. Okay, so we're looking for what goes with U. So when you use the codon table or the wheel, you can use whatever you want. I will say on the AP test, they always give you the table. So you don't need to memorize this at all. They just give it to you. You just need to know how to read it. So when you read the codon table, you always start here on the left. You find your first letter. We start with U, and then you find where the second letter, U, and then this would be your third letter. So it goes first letter, second letter, third letter. So U, right here. Go away, previous. That represents phenylalanine, and then its symbol is F. So here, U is going to be phenylalanine. Okay? So we don't look for, this is where people get crazy. You do not look up the anticodon to find the amino acid. It's called a codon table because the codon determines the amino acid that is carried. So then our next one we're looking for is ACG. Try to find it. You start here first. A, you find where it intersects. C, G. So this would be threonine, or designated as T. It's hard to write and hold my phone at the same time. Here's how to use the wheel. We'll do a last one with the wheel. We're looking for CUG. Wow, I need to paint my fingers. So with the wheel, you just start in the middle and then you work your way out. So I have C, U, G. It's leucine, or designated as L. Wonderful. So that kind of shows you just what everything is. You'll also see here we have our stop codons. They're designated with negatives here. This is our start codon, methionine. It kind of shows it over here, or not. Kind of. This is our start codon here, and these are our 3 stops. Ooh-ah, ooh-ag, ooga. Cool. Another cool thing you can kind of see is there is redundancy. So to get serine, I could have any of these different sequences. So when we talk about mutations in the next, over here too. So when we talk about like mutations in our next video, it's possible that I could have a mutation, but it doesn't actually change my protein because of the redundancy within the codon table. So that allows there to be mistakes in our DNA replication, but not have the mistake carry through into our actual protein. Cool, so now we have our different amino acids. Stop codons actually don't carry like an amino acid. As you saw in the codon table, it just says stop. So they'll just be a tRNA that bonds here, but it doesn't carry an amino acid, so it just kind of stops the chain. So for all of these things to come together, they all kind of come together at the ribosome. So we're gonna draw a ribosome. A ribosome will bind first at the start codon, and a ribosome has 2 subunits. I'm gonna try to draw this so it all makes sense. Okay, so the top subunit is the large subunit, like so. And then it has another subunit that's kind of smaller. Maybe pick a color that you can read through. So this is our ribosome. Cool. So let's kind of talk about ribosomes for a second. Ribosomes have 2 subunits. They have one large, which is usually the top portion, and one small. Okay. Both subunits are made of the two of the same components. So every ribosome is made up of 2 things. It's composed of rRNA, which is ribosomal RNA. So when we talked about ribozymes way long ago, these are the ribozymes. So these are the portions of this ribosome that are help catalyzing this reaction of bonding the amino acids together. So it's made of ribosomal RNA. And then let's just make a note so you remember these are ribozymes. And then it's also made of protein. Why didn't I write that anywhere? Okay, so it's composed of rRNA, which is these ribosomes. They catalyze the production of our polypeptide, and it's just made of straight up protein. So it's a complex of protein and like RNA wound together. And then pieces of the RNA will be these catalysts that catalyze the production of our polypeptide. Wonderful. Okay, on this ribosome, on the large subunit, there's actually 3 different binding sites. I usually just draw them as cute little humps. Let me find a color that can draw over certain things. Okay, so where we have our start codon, that will be in our first binding site. There'll be a second binding site and a third binding site. Each of these binding sites can bind a codon. So we can have 3 bound here, 3 bound here, 3 bound here. They bind in sequence though. So this is binding site number 1. So this one's always first, and it's called the A site. Then, so our codon will start here. And then what ends up happening is your ribosome will slide. So the ribosome will move, and then the AUG will be here now. So it'll bind here second. And this is called the P site. And then the ribosome will move again. So it's kind of like ticking along the codons. And every time it ticks down 1, your little codon here will move down 1 spot. So then it'll finally end up here. This is called the E site. And then it won't be bound to the ribosome or like within the ribosome anymore. So let's kind of talk about our 3 sites. So we have first the A site. The A site accepts the tRNA. So the A stands for accepts tRNA. So we have these free tRNAs. The only time this tRNA can bind to this codon is when this codon is in the A site. So now that AUG is here, this tRNA would come over, it would match up its anticodon to this codon, and then it would drop off its amino acid right here. Okay, let me say that again. So when a codon is in the A site, the tRNAs will be chemically attracted to it. The codons and the anticodons, they want a hydrogen bond with each other. So the tRNA will move over, they will bond, and then the RNA, the ribozyme on the ribosome, will take off, cleave this methionine, and it will put that methionine on itself. So it'll kind of grab it and hold it for itself. So let's just draw that methionine kind of chilling here. Okay, so now what you would have is you would have this like tRNA here that doesn't have methionine anymore because it's now bound to the protein. Our ribosome's gonna move over, it's gonna click. Now AUG is here at the P site. When it's at the P site, this is like technically when the polypeptide is being formed. So I kind of went over everything in one step. It's like a two-step process. So the tRNA will bind. We're gonna cleave our amino acid here, and then we're gonna bind our amino acid at the P site. So we're starting to form our polypeptide up here. Once the ribosome moves again, we kind of tick over once more. Now AUG is here at the E site. This is when the tRNA will exit the ribosome. Okay, so that tRNA will have its amino acid cleaved, it'll bind to the ribosome, and then it will leave the ribosome. So everything's moving at once, okay? So you kind of have to imagine this. When this is here, we're gonna have methionine in the A site. So we're gonna then tick over one, and then we're gonna have a new tRNA bind. Whatever is in the A site, that tRNA is gonna come over, bind. We're gonna cleave off its amino acid. Once we move it over again, and it's in the P site, then that amino acid will get bound here as a chain. If you move it over again, so it's in the E site, the tRNA will leave. So we're kind of just starting to add these tRNAs one at a time. We start to cleave their amino acids off, and then we bond them up here one at a time. What you end up getting, so if methionine is here, it's the start of our chain. Once we add another amino acid, this methionine will move up, and the new amino acid will kind of be peptide bound right below it. Okay, I hope that all made sense. So you're gonna end up with a protein. Okay, remember we said we have 1, 2, 3, 4, 5, 6, 7. So you're gonna end up with 7 amino acids. Each codon codes for 1 amino acid. The start will be methionine. And then in our sequence, we had phenylalanine. Then we had threonine. Then we had leucine. And then I didn't look the others up, but I can. So leucine went with here. So then we have to do UCG, which is leucine again. GAC, asparagine. That's the one that makes you able to spell the asparagus smell in urine. AAG is lysine. And that would be our amino acid. So we've made a protein. Well, we have a polypeptide, so this is like primary structure. So the only thing that needs to happen post-translational modification All that needs to happen is we need to have this protein to be folded. Remember, so it's going to fold at least to tertiary structure, and then it could get to the optional quaternary structure. Okay, I wanted to show you a diagram. Oh, we're going on another adventure. Don't drop your phone, don't drop your phone. I did it. That was the smoothest I've done it so far. Okay, so I wanted to show you this because there's a few things here. So first here, so this is what we just went over. So here we have our DNA. This is transcription. We're growing our RNA. We've processed it. Here's our mature mRNA. It'll travel outside the nucleus. And as you can see, you can actually have multiple ribosomes making proteins at the same time. So we just drew one, so like maybe this is the one we drew, but once that start codon is free again, another ribosome is going to attach, and so you can create multiple proteins at once, which is super nice, because eventually you're gonna start getting those ribonucleases cutting into your RNA, and then you're gonna get incomplete proteins. In prokaryotes, because they don't have a nucleus, it goes a little bit differently. It's a little faster actually, so they're smaller in size, but so we're gonna have the same two steps. You're gonna have transcription, but you can kind of see here, we're finishing transcription. We don't need to do post-translational modifications because we don't need to exit the nucleus. I don't think prokaryotes have as many ribonucleases as eukaryotes, but I need to look that up. Transcription actually occurs at the same time as translation. So while transcription occurs, we create that start codon down here. Ribosomes start attaching and creating proteins even before the mRNA is done being transcribed. And then once the mRNA is done being transcribed, they can kind of finish off and create these proteins. And it kind of just carries out protein synthesis a lot faster because they don't have a separation between the cytoplasm and the nucleus. So they don't need to have this like modification that kind of like transports it out of the nucleus. Okay, we've made a protein. So this was our end product. So remember, this sequence of amino acids originated from this sequence of DNA. So your DNA is what carries the instructions to create all the proteins in our body. So when we start talking about genetics and stuff like that, like why do I have black hair versus somebody having blonde hair or red hair, my genetic code has a code for a protein that when it folds is dark. Or people who are blonde just lack this protein, so they just don't have it, or they don't have as much of it, so it's not as saturated. And then there's different color pigments, so then you can get red types of hair and then different shades. But a lot of our features are usually due to like the presence of a protein or the absence of a protein, or it could be a variation of the protein. So we'll kind of talk about that when we get to genetics. All right, come back and watch my mutation video. Bye, guys.